Hello everyone, this is our group assignment video on food microbiology. The title is To Spot the Spoiled Milk. In this video, our group will demonstrate on how the methylene blue reduction test can be used to detect the milk spoilage. Milk is a highly nutritious food that contains proteins, fats, vitamins, and sugars, which also make it a suitable environment for the microbial growth. When the bacteria grow in milk, they break down the lactose into lactic acid and produce unwanted changes in smell, texture, and appearance. As spoilage continues, the milk quality will decrease and may become unsafe for consumption. Therefore, methods such as MBRT are very important in food microbiology to evaluate the milk quality. The methylene blue reduction test works by using methylene blue dye as an indicator of microbial activity in the milk. Methylene blue in the sample containing microorganisms get reduced to leucol or colorless at the cell surface via the transmembrane reductase enzyme, which are present in the cell membrane of the microbe. When the microbes are actively growing, they will consume the oxygen in the milk and reduce the methylene blue dye, causing the blue color to disappear. A faster decolorization indicates a higher bacterial activity in the milk and poorer milk quality. On the other hand, milk that remains blue color for a longer period of time indicates a lower microbial growth and a better quality of the milk. The objective of this experiment was to investigate the effects of different storage time and different storage temperature on microbial spoilage in UHT low-fat milk using the MBRT method. We compared the milk stored at refrigerated temperature, which is 4 degrees Celsius, and at room temperature for both 24 hours and 48 hours. We also compared the samples with a control sample. Through this experiment, we were able to observe the changes in milk quality based on their color, smell, texture, and appearance.
Hi, my name is Kwon Chi-Xuan, and I would like to talk about the materials and setup in this experiment. The materials used in this experiment included UHT low-fat milk samples, methylene blue solution, sterile test tubes, micropipette tips, measuring cylinders, and a test tube rack. We also used a water bath set at 37 degrees Celsius and a Bunsen burner for aseptic techniques. Proper aseptic handling was important to prevent contamination during experiment. Gloves were also worn throughout the experiment as a safety precaution. First, the test tubes were labeled according to storage temperature and storage type. Next, 10 ml of milk sample were transferred aseptically into sterile test tubes. Then, 1 ml of methylene blue solution was added into each tube. The tubes were closed tightly and inverted several times to ensure proper mixing of the dye with the milk samples. After that, all tubes were placed into a water bath at 37 degrees Celsius for incubation. After that, we store the sample at room temperature and 40 degrees Celsius for 24 hours. During day 2, we are doing the control preparation, and after this, we will add the methylene blue to all the sample. One milliliter of methylene blue solution was added into each tube. The tubes were closed tightly and inverted several times to ensure proper mixing of the dye with the milk samples. After that, all tubes were placed into a water bath at 37 degrees Celsius for incubation. After adding the methylene blue, we'll incubate it into the water bath at 37 degrees Celsius, and after this we'll observe the results. Now, let's talk about the observation process. The milk samples were observed after 30 minutes, 2 hours, 6 hours, and 8 hours of incubation. During each observation, we checked whether the blue color had disappeared completely or if a faint blue ring remained at the top of the tube. We also observed physical characteristics such as texture and appearance of the milk. Throughout the experiment, the samples were gently inverted to redistribute the cream layer and ensure accurate observation. Proper handling was maintained to avoid disturbing the results.
All UHT low-fat milk sample remain blue after 30 minutes, 2 hours, and 6 hours of incubation. This indicates a negative MBRT result, meaning a milk sample was still in good quality and had low microbial activity. No sour smell was detected in any samples, and the milk texture remained smooth without curding or clot formation. However, the 48-hour milk sample showed a slightly lighter blue color compared to the 24-hour samples, suggesting a slight increase in microbial activity over time. From our experiment, the 48-hour UHT low-fat milk exhibit a higher degree of decolorization compared to the 24-hour UHT milk. This suggests that the quality and stability of the milk decrease with longer storage duration. The greater decolorization observed in 48 hours may be due to increased microbial activity over time, resulting in a reduction of the dye. In addition, refrigerated samples show better stability compared to room temperature samples because low temperature slow down bacterial growth. Therefore, storage temperature and duration both affect the rate of milk spoilage. Try high temperature processing heat dairy product from 275 Fahrenheit to 300 Fahrenheit for 2 to 5 seconds, similar to ultra pasteurization. This renders products commercially sterile and shelf stable. High temperature short time pasteurization heat product about 161 Fahrenheit for 15 seconds, eliminating harmful bacteria to align with PMO milk requirements. Products are not shelf stable, refrigeration is required. Low temperature long time pasteurization keep dairy product to around 145 Fahrenheit to 150 Fahrenheit for 30 minutes. This process eliminates harmful bacteria while preserving product quality, but refrigeration is required. Temperature favors to moderate microbes such as Clostridium and Bacillus. These are spore-forming microbes, especially Bacillus sporothomodurans that are extremely heat-resistant and become a major cause of spoilage for UHT pasteurized milk. In conclusion, the methylene blue reduction test was successfully used to evaluate microbial spoilage in milk sample. The results show that UHT low-fat milk remained in good condition throughout the experiment. Longer storage time and higher storage temperature increase the possibility of spoilage. To prevent milk spoilage, milk should be stored under refrigeration and handled hygienically after opening. Overall, this experiment helped us understand the relationship between microbial growth, milk spoilage, and storage condition.